Hello, a warm welcome to you from SGT University. I am Dr. Nimapreet from Faculty of Medicine and Health Sciences. So today we'll talk about cellular immune response. Development of cellular immune response. Cellular immunity refers to specific acquired immunity accomplished by the effector T cells and macrophages. It is also called as cell-mediated immunity. The role of cellular immunity is that it protects the host against fungi, most of the viruses, intracellular bacterial pathogens like Mycobacterium leprae, Mycobacterium tuberculosis, and Brucella. It participates in allograft rejection and graft versus host reaction. It participates in delayed hypersensitivity reaction. It is also associated with certain autoimmune diseases. It provides an immunological surveillance and immunity against cancer, that is, tumor immunity. What are the types of cellular immune response? Like the humoral immune response, the cellular immune response is of two types: primary cellular immune response and secondary cellular immune response. Primary cellular immune response, which is produced by contact with the foreign antigen. Secondary cellular response, which is produced when the host is subsequently exposed to the same antigen. The secondary cell-mediated immune response is usually more pronounced and occurs more rapidly. Further, because of the availability of specific memory cells, an increased number of effector cells are produced in secondary response. Let's talk about the different phases of cellular immune response. First stage is antigen processing and presentation. Recognition of antigen by the lymphocytes. T-lymphocyte differentiation. Attack phase of cell-mediated immunity. Inactivation of antigen. So coming to the first stage, that is antigen processing and presentation. Antigen after entering the host body is phagocytosed and is degraded into polypeptide fragments by the antigen-presenting cells or the antigen-processing cells, which include the macrophages and the dendritic cells present in the peripheral lymphoid tissues. The antigen polypeptide fragments then becomes associated with the MHC antigen and they are expressed on the surface of the antigen presenting cells. There are two modes of antigen processing. Processing of the phagocytosed material. For example, if the antigen is bacterium, this is accomplished by combining with MHC class II antigen. Second is processing of the antigen derived within the cell. For example, if the antigen is viral antigen that is synthesized in the infected cell by combining with the MHC class I antigen. Second is recognition of the antigen. Recognition of the antigen is T-lymphocytes. They possess the antigen recognition T cell receptors known as the TCR receptors. These receptors, they serve as specific surface receptors, recognizing only one single antigenic determinant on the antigen being presented to the lymphocytes. Further, mature lymphocytes can be differentiated into two antigenic subtypes depending on their surface antigens. They are CD4 cells and the CD8 lymphocytes. CD8 cells recognize the combination of foreign antigen and class 1 MHC antigen. CD4 cells, they recognize the combination of the foreign antigen and class 2 MHC antigen. Third stage is the T-lymphocyte differentiation. CD8 type of lymphocytes, after combining with the MHC class 1 complex, they are differentiated into cytotoxic T cells and the suppressor T cells. So, CD8 T lymphocytes, they differentiate into cytotoxic T cells and the suppressor T cells. So, cytotoxic T cells, they are known as TC cells, while suppressor T cells, they are known as the TS cells. Now, the CD4 type of lymphocytes, they combine with the foreign antigen MHC class 2 complex and they differentiate into helper T cells known as the TH cells and the delayed type of hypersensitivity cells known as the TD cells. There is a terminology known as T-T cooperation, that is the differentiation of T lymphocytes into T helper cells, into the cytotoxic cells, suppressor cells and the delayed hypersensitivity cells is interdependent. So this conversion is interdependent. This interdependence is known as T-T cooperation. Then occurs the release of the differentiated T cells. The differentiated T cells, that is, which are formed, are released into the lymph, then enter the blood through which they are distributed throughout the body. They also pass out through the capillary walls and enter in the tissue fluid. From the tissue fluid, they enter back into the lymph and then into the lymphoid tissue and once again in the blood. Thus, T lymphocytes circulate again and again throughout the body, sometimes lasting for months or years. T lymphocyte memory cells, they are also formed. Then comes the attack phase of cell-mediated immunity. Here comes the role of two types of cells, that is the cytotoxic T cells and the natural killer cells. So they both are responsible for the attack phase of the cell-mediated immunity. Cytotoxic T cells. Cytotoxic T cells, they are the effector cells in the cell-mediated immune response. They recognize the fragments of the foreign proteins produced by the infected cells. They possess the accessory protein that binds to the class 1 MHC molecules. The activated cytotoxic T cells secretes the proteins and that disrupts the membrane of the target cells and trigger the apoptosis. So in this way, you can see the cytotoxic T cell when it recognizes by binding with the class 1 MHC molecule. So by the release of the perforin and by release of the various proteins, it causes the lysis of the cell. The various mechanisms by which the cytotoxic T cells destroy the antigen-bearing cells are by perforin-mediated killing. Here the cytotoxic T cells, after binding with the target cells, secrete the hole-forming protein known as perforin. The perforins literally punch round holes in the membrane of the target cells in the presence of the extracellular calcium, that is the calcium-mediated lysis. The pores so formed cause the cell death by disrupting the cell homeostasis. Second mechanism is by lysis by the cytotoxic substances. After binding with the target cells, the cytotoxic cells enlarge and they release the cytotoxic substances. Third mechanism is apoptosis, that is also known as programmed cell death. Here the cytotoxic cells secrete the tumor necrosis effector beta. This increases the calcium permeability of the antigen-bearing cells. The increased intracellular calcium activates the enzyme that causes the degradation of the nucleus, producing apoptosis.
Let's study about the role of helper cells and the suppressor T cells in the cell-mediated immunity.
Helper T cells are of two types, Th1 and Th2. Helper Th1 cells play their role by secreting the three cytokines, that is interleukin 2, gamma interferon and tumor necrosis factor beta. Role of helper T cells, that is Th2 cells, they secrete interleukins 4, 5, 6, 10 and 13. Cause the activation of the B lymphocytes to produce the antibodies. Then comes the role of suppressor T lymphocytes. Suppressor T lymphocytes regulate the activity of cytotoxic T cells. Thus, cellular immune response is a balance between the cytotoxic T cells and the suppressor T cells. Suppressor T cells also play an important role in preventing the cytotoxic T cells from destroying the body's own tissue along with the invading organisms. So, suppressor T cells also suppresses the activity of helper T cells. So, this was about the cell-mediated immunity. Now, let's summarize the differences between cell-mediated immunity and the humoral immunity. Cell-mediated immunity, that is mediated by T cells, while humoral immunity is mediated by B cells. Antibodies are not formed in cell-mediated immunity, while in humoral immunity antibodies are formed. Here, the T cell receptors bind to T cells and then bind to the antigen, while in humoral immunity, B cells produce antibodies and antibodies bind to the antigen. Cell-mediated immunity protects against intracellular pathogens, while humoral immunity protects against extracellular pathogens. The cells involved in cell-mediated immunity are macrophages, helper T cells, natural killer cells, and cytotoxic T cells. Over here, T lymphocytes, B lymphocytes, and macrophages are involved. This mediates type 4 hypersensitivity reaction. These mediates type 1, type 2, and type 3 hypersensitivity reaction. In cell-mediated immunity, both CD4 and CD8 T cells are involved, while in humoral immunity, only T helper cells are involved.
So today we covered cellular immune response, its various phases and role of cellular immunity in our body. So next time we come, we'll explore another interesting topic in physiology. So till then, keep learning, keep growing. See you next time.